Hello, I'm Dr. Jennifer Jackson. I'm a registered nurse and assistant professor in the Faculty of Nursing at the University of Calgary. And this video presents an overview of quantitative research. So here in part one, we're going to talk about general principles of quantitative research, approaches, big ideas. In the next video, we'll talk about specific designs and how those are implemented. So we'll get started. So should I do a quantitative study? What kind of study should I do? So for researchers, the first question is, what type of question do you want to answer? What is the problem? What is the thing you want to understand? So you don't start from saying, I'm going to do a correlational study and let me find a problem. You find a problem and then from there decide what method is going to be most appropriate. And now there's not to say there's a single method that is the right way to do research. You can have topics, especially things like mental health, like anxiety. You can measure the prevalence of anxiety, but you could also understand the experience of anxiety. So there's more than one right way to understand a topic. It's just what are your aims and what do you want to achieve with this? You need to make sure that your problem drives the methodology and not the other way around. So sometimes novice researchers want to say, I'm going to do a survey and I'm going to do this and I'm going to ask people about this. And the bigger question is, what kind of problem do we want to solve? Does that problem lend itself to a quantitative study? So what does quantitative research do? Generally, it tests hypotheses. So this uses a deductive approach. So we come up with a problem and we might say something like, we know that people are experiencing high levels of anxiety before surgery. We need to do something about that. So then we can look and say, okay, maybe we want to offer people music that they can pick to listen to while they're in the waiting area before they go in for surgery. Maybe that will decrease their anxiety. So we can test and see if that works. Quantitative research allows us to test interventions to see if they achieve the desired outcome. We see quantitative research very extensively in health, particularly around anything related to drug trials or novel treatments, approaches to managing symptoms. All of that has a pretty significant quantitative component. We'll look at differences between groups and also consider how the relationships exist with variables. So maybe we find that offering music before surgery is really helpful for people under 50. However, people over 50 struggle to use the their phone or whatever it was to play the music. Maybe we had to have some kind of sterile generic phone and they struggled with that and they got distracted and it didn't achieve the desired effect. So we can look at, okay, well, was there an age divide? Was there a gender divide? What was happening among different variables here so that we can understand, was this effective or not? And was the effect due to chance or did we actually measure something that was happening here? We also want to look at how things change over time. So we see this a lot, especially with population level prevalence data. So what are the rates of COVID-19 in a given population? That is a perfect quantitative problem to look at. How does this rate change month to month, day to day? Is it different in the wastewater versus looking at, nasopharyngeal swabs? What about vaccination rates? How did that affect it? So we can look at all these kinds of metrics. What we're determining is whether these events were due to random or potentially unknowable factors in the environment, or do they exist because of a specific association between variables or because of a specific intervention we conducted? So the biggest difference between quantitative research and qualitative research is in quantitative research, you control the context so that you understand what has caused an effect. In qualitative research, there is no control. You deal with the full messiness of the context and you understand something in its crazy environment. So that is the biggest difference, is can you control an environment to try and isolate an effect, or are you dealing with the environment as it comes and how things happen within that environment? Okay. Another consideration within at least descriptive studies for quantitative research is we have retrospective and prospective. So retrospective is we look backwards at something to see how its effect has happened. So prospective, we know we're going to start something and we look forward. So what I mean here is retrospective, none of us knew the COVID-19 pandemic was coming. So there was no way we could set up pre-pandemic studies to then watch how the pandemic unfolded. So, I mean, we got there later, once people got established doing research in the pandemic. But initially, we're looking back. If you have, say, a natural disaster, you're gonna look back at what happened and evaluate the consequences of that. Prospective is particularly common with any kind of health treatment. So we are going to start giving people this medication and see if it makes a difference. And so our study is set up before the intervention occurs, and we're going forward to assess this intervention. So you'll see the terms retrospective and prospective. And so it relates to where you place the study. Did the researchers get set up before they got started and they could observe it while it happened, or are they looking back after the fact? One of these isn't better than the other. It's just what is practically possible in the environment based on what you're trying to understand. So we talk a lot about variables in quantitative research, and we have three main kinds of variables. Variables are the Lego blocks that build our hypotheses. So if our hypothesis or hypotheses are the little Lego car, the variables are each component of that car, each little Lego brick that's part of it. So an independent variable is what the researcher can control. So this might be something like the medication administration in a drug trial. The independent variable is whether the person got the medication or got a placebo. And that is something that we can control and manipulate. And I mean manipulate in the scientific neutral sense of the term. A lot of this language came in a separate lecture. We can unpack how this language evolved over time. And so some of it can be quite loaded in our modern context. But with all these things, I mean it in a scientific context. So we can manipulate the environment. We can give some people a medication and not give it to others. And that's our independent variable. The dependent variable, this is the outcome. And this is what we have no control over. So we want to see, does this medication make a difference for people? If it does make a difference, is that difference positive? Is that difference what we think it will be? Are there unexpected differences that come forward? And is this difference something like a positive association, a negative association? And when we use the terms positive and negative here, we're talking about positive, the number goes up, negative, the number goes down. That doesn't necessarily mean positive is good and negative is bad. You could have, give a medication and have a negative relationship with pain. That would be a good thing in that the medication helped decrease pain. So the dependent variable is the thing that you want to measure and see what happened when we did our intervention? Was this drug effective? We could also have confounding variables. So we can predict a direct relationship between our independent variable and our dependent variable. However, there's other stuff that might get in the way. And a confounding variable is something we want to be aware of so we can take it into account. So a confounding variable is something that might interrupt or interfere or change this path between the independent and our dependent variable. So a confounding variable might be something like having diabetes. So we could have, okay, we're administering a medication, we're giving it to one group of people, we're not giving it to the other. However, we found that people with diabetes respond to the medication differently. And so that's a confounding variable that we want to take into account when we're doing statistical analysis so that we can truly say, yes, our independent variable caused our dependent variable, rather than, well, this other factor was in the mix and now we don't have a hypothesis we can test anymore. So we go into the study, deductive approach, we have a hypothesis in mind. So this medication will decrease pain. And then we test that by controlling the context of the study. So we give the medication to some people and not others. We can statistically take into account, okay, people with diabetes might have neuropathies, and so this medication might work differently among that population. So there's all kinds of ways that we can statistically take into account these different things, but we need to be very clear on the hypothesis that we are testing before we start a relationship. When I'm appraising research, that's one of the things I look for is their proposed hypothesis clear and well articulated? And if it's not, then that's a big red flag. Because you can have a study that says we're looking to test the effect of this medication on pain. Okay. But then later in the paper, if I see, does this medication have an association with an outcome around pain, then I get nervous because those are two different things. And you need to make sure that the researchers are very consistent and they know what they're doing. So we have variables. These are the Lego blocks that we want to measure, but we know we have different kinds of Lego blocks. So how do we think about these different Lego blocks? So variables, generally, there's three main types of measurement, and they break into different categories depending on the nature of the thing that you're measuring. So nominal variables are measured and based on name. So you count the number of groups. So a nominal variable, a classic one, is gender. So you have men, women, and people with diverse gender identities. And you can set that up so that one group is not better than the other. You're simply counting the number of things that you have there. There's also no hierarchy of the variables. What's different is the name that we give them, hence nominal. So you could also look at, like, what city are you from? Calgary, Edmonton, Vancouver, Regina. You're looking at the named variable and there's no difference between them. Okay, so nominal variables are typically used to describe the sample in the study. How many people did we have from this location? How many people did we have from this gender? How many people did we have who work part-time versus full-time? Things like that. So those variables are separated by name. Ordinal variables do have a hierarchy, but they are not necessarily equal increments. So where we often see ordinal variables is tell me your income. And it might be under $50,000, 50,000 to 75,000, 75,000 to 100, 100 plus. Well, the difference between 0 and 50 and 50 and 75 is not equal. But that type of measurement might be helpful because maybe your cutoff is the poverty line. So what you're asking somebody is, are you below the poverty line? Are you considered just above the poverty line, way above the poverty line, that kind of thing. So this is also useful when it is maybe challenging to get an absolutely precise number. So if you ask someone for a range of income and say about how much money did your household make last year, you could probably pick up and say, you know what, we're in this category. But if I said, give me a dollar value for exactly how much money your household made last year, this would be really difficult to do. So ordinal variables are a way you can get a sense of something like, are you above or below the poverty line, without making your participant kind of work too hard if the exact precise dollar amount is not particularly necessary to understand your analysis. Finally, we have ratio level variable, and these do have a hierarchy, but the difference between each item is exact. So $1 to $2, there's exactly $1 difference. $2 to $3, there's exactly $1 difference. So we know that it's very precise because there are exact fixed increments that are the same across the whole scale. So an example of ratio level variables in health, that could be weight. Is this person with dialysis? What is their weight at, and how do we calibrate their treatment accordingly? So when we get into looking at statistical tests, there are entire university degrees on statistical tests. So I'm providing necessarily a very brief overview. But what you do is, was this statistical test appropriate for this type of research study? You look at what variables, what is their independent variable, what is their dependent variable, and what type of measurement was it? So did they have a ratio level independent variable and a ratio level dependent variable? So then there's many tools available that can help you out with this. But I would pull out my Paulett and Beck and look in the chart and say, okay, ratio level independent, ratio level dependent, the appropriate test for that is a correlation. So when you're figuring out, did they use an appropriate test, you want to look at the level of measurement and the type of hypothesis they're testing, and do all those pieces fit? Generally, we have descriptive tests and inferential tests. So descriptive tests tell us who did this intervention or who was part of this study. Descriptive, describe what's going on. So in every research study, table 1 usually is the first part of the results is always the table that has the descriptive information about the participants. So how many men, how many women, how many gender diverse people, how many people work full-time, how many work part-time, how many work casual, those kinds of things. So that information is presented right up front so we can make a judgment about how representative or not that population is. Then we can look at inferential statistics, which are, we can make inferences based on those kinds of statistics. So this is where we get correlation, regression, ANOVA, t-test, chi-squares, all of these different types of statistical tests. And I don't expect you to be able to memorize all of them. What I want you to know is this describing or is this telling us about a relationship between the variables? And is the test appropriate for the types of variables and the types of hypothesis we have and the level of measurement for each of those variables? So one thing we need to do is, when we're appraising research, is to say, did they tell us the whole story? So when we ask, are the outcome data complete? We're not looking at the participant side. We're not asking, did this person go through all of the questions? Because as a participant, they have a right to not answer questions. So we're not asking about participant like completion of data. What we're asking about is researcher reporting of data. And so they need to report all of it, not what's called cherry pick. So if they say we are measuring 5 variables, I want to see 5 variables reported in the analysis. I've seen where marketing or branding things will say, look at the difference, between A and B, A and B, A and B, and then, oh, here's an A and C. And making it as though it was consistent, and you can say, no, they snuck that in there because they are cherry picking the data. So if you look at like the Nike marathon shoes, well, you could say, yeah, they give people the fastest times, they're the most streamlined shoe, they're comfortable, they make you run faster, but they also disintegrate over time and they're good for like 2 races tops. So Nike might not want you to know that. So they might hide those data and say, oh yeah, it just makes you run faster and they're so great and all of this. But if I'm a researcher, I also want to know that those shoes fall apart almost instantly so that I can make an informed decision about, okay, if I'm promoting running among a population, like, are those shoes going to be part of an effective intervention to help people run faster? You want to see that there's full accounting for the variables. So if they say they're testing hypotheses with 4 variables, you need to see 4 variables in the results. I've got p-values here, but we're going to get into that a little bit later. So what kind of samples do we want in quantitative research? Generally, we want larger numbers relative to qualitative research. So qualitative research, you can have a perfectly good study that might have 6 or 8 participants. Depending on the methodology you're using, that might be completely appropriate. For a quantitative study, you might have 6,000 or 8,000 participants. And so generally you want larger numbers. It depends on what type of study you're doing, how large those numbers exactly are. For a randomized controlled trial, you might have like 35 and you might have 70 people with 35 in the control group and 35 in the experimental group. However, if you're doing an epidemiological study, you would expect like tens of thousands across multiple countries. So generally the numbers are bigger, but it depends on what you're trying to measure and how intensive the study is to determine exactly how many people you need. Population is randomly selected. So you could walk out and say, I'm going to take 30 random people and have them do an intervention and see what the outcome is. So this is different than qualitative research where we pick people that have specific characteristics or specific experiences we want to understand. Whereas here we want a representative group of the population. We want the people in the group for our research to reflect the people who are out in the world so that we can draw conclusions. So one of the biggest challenges with a lot of social science research is that the studies are conducted at universities. And so the main populations are like 18 to 20-year-old white upper class students. So you run into a lot of, they try to draw big conclusions, but it was measured on this very specific population. However, we do have, when we say randomly selected, it's randomly selected within our sampling frame. So we do have some studies that are genuinely like random groups of people. However, if our sampling frames, we want to understand outcomes among nurses, we will be picking a random group of nurses that reflect the population. So if we do that, our rate of women in that study is probably going to be 88 to 92%. That doesn't mean we did the study wrong. That means within the group of nurses, which is called our sampling frame, if you think of like a picture frame, the picture within that frame, 90% of nurses worldwide are women in just about every country in the world. So if I see a nursing study that's say 50% men, 50% women, I'm going to be nervous because that's not what a nursing sample is. A nursing sample is 90% women anywhere in the world. So it has to reflect the population, but that doesn't necessarily mean it has to be equal. And so we're looking at these samples for characteristics of age, gender, race, and depending on the question you ask, there might be other factors like household income, which is a proxy measure of socioeconomic status. Are you above or below the poverty line? Things like that. So we've got our group of people. Now we need to get some information from them. So we may use surveys. Surveys are a mainstay of quantitative research, especially if you were asking the participant to give you their opinion or to provide the input. So you give someone a survey, they fill out the questions, then you take those answers and you analyze them. When you are doing surveys, you generally want to use what's called a validated tool. So this is a scale that is something that has been widely tested and it is known to be an effective measurement of this thing. So for example, we have the Connor-Davidson Resilience Scale. This is a scale that has been created by Connor and Davidson, and they have different versions of it. There's 20, I believe it's 20 questions, 10 questions, and 2 questions. And they have tested those questions among many groups of people. And when I say tested, what I mean is they gave the survey to people of all genders, people of all races, people of all socioeconomic status, all different geographic areas. And they found that this tool measures resilience across all of those groups. And it might need tweaks here or there, it might need translation, but generally it is known to be a very effective tool. So that is what you want to use in your survey, because then if I do an assessment of nursing student resilience, I can then compare it to all these other studies that have used the same tool. Whereas if I come up with my own questions, I may or may not actually measure resilience. I might accidentally end up measuring something like self-efficacy, and I can't compare it to any other studies. So you want to know what kinds of tools would I expect to see here and how are they being applied. It doesn't mean it has to be an exact carbon copy from one to another. Sometimes tools are modified based on the population or the setting, but they're modified very slightly because it's like using a thermometer. We can check the same thing all the time. We also have different kinds of verifiable measurements. So you could be doing chart reviews and look, every time it was documented that someone experienced pain. So that is a kind of objectively verifiable measurement. It doesn't matter who's looking at that chart. If pain is documented, pain is documented. With any kind of research, the data collection tool is not a determinant of what kind of study you have. So like participant observation is a mainstay of a qualitative method called ethnography. You can also use observation in quantitative research, but the intent is different. The intent would be count the number of people who use their phone while they're waiting for a light to change at an intersection. Or interview people and ask them about how many times they have bumped into someone because they're using their cell phone and they're not walking or watching where they're walking. So the data collection method itself doesn't tell you what kind of study that you're looking at because methods can be applied a bit differently. But you can tell looking through like, okay, what were the variables? What was the level of measurement? What hypothesis is being tested? Then you can look at the data collection method and associate how is this related. So if the hypothesis is looking at pain, you can look at, okay, we're doing chart reviews and we're assessing every time pain was documented. So don't get caught up on, oh, they did interviews, it must be a qualitative or a quantitative study. It's more important to look at the data collection method within the big picture. And so here for the end of part one, we're going to do a very brief discussion of data analysis and quantitative research. And again, there's like entire degrees based on this. This is just a very high level overview. So quantitative research is heavy on the front end. You have to put a lot of work in up front to say what kind of scales am I using? How am I going to recruit people? How am I going to sample this population? And so it's really, really heavy on the front end. But once you get your data analysis set up, push the buttons and the computer spits out some numbers, you interpret them, and you have an answer. So you have an outcome related to your hypothesis. In contrast, qualitative research is very heavy on the back end. So you doesn't take a lot to get set up, but when you are collecting data, say doing interviews and coding, that is extremely labor intensive. So when we're looking at analysis that's quantitative, the researchers are going to be pulling their hair out in the first parts of the study. So generally, quantitative analysis is done with software. Back in the day, you know, poor Florence Nightingale kept lists and manually calculated statistics, but today it's done with software. So generally, you have programs like SPSS, Python, R. There's many different kinds of them, and they work depending on what you want them to do. So you put in, these are the scores for my independent variable, and these are the scores for my dependent variable. And then you get the software to test the hypothesis. If you want to do really fancy stuff, like modeling and machine learning and all this, you can write your own code for it. That's about the limit of my knowledge on that subject. So I encourage you to take advantage of the other resources that are out there, because there's lots of great research happening with machine learning and modeling and all this. So you hit the analyze button or equivalent on your software. It'll test your hypothesis and it'll spit out some numbers. Now, generally, this is at a very high level, but generally it will tell you what is the relationship between these variables. And if you're using what are still considered quite standard approaches, you'll get a p-value associated with your numbers. So if you're looking for Bayesian statistics, that's again a whole other thing. So there's different techniques for analyzing these data, but generally, if you get a p-value is a pass or fail. It does not tell you anything about the nature of what you've measured. A p-value tells you, was this due to chance, or can we be confident that we actually measured an effect? So if you have a higher p-value, so generally the pass-fail threshold is set at 0.05. If you have a p-value that's higher than that, you say, you know, maybe my resilience score was higher among nursing students and engineering students, but it doesn't pass the p-value test, so I have to assume that these data are random, or this measurement is a fluke and it's due to chance. And if I measured 5 more times, I would get different results. So I can't have confidence that I've actually detected a statistical difference between these two groups. However, if your p-value is lower, if it's under this threshold, it means that, yes, I have confidence that what I have measured is actually a relationship that exists out in the world. And now I can look at what are the differences between the scores and make inferences based on that. So if your p-value is higher or lower, for our purposes, it's just a pass-fail. So you don't say, oh, wow, this is really, really significant. You can maybe have more certainty that it's not based on chance, but it doesn't mean that, like, oh, the scores are going to be way higher or something like that. So this is something that tricks us up when we're learning about it. So be sure to know that a p-value is a pass or fail. in most health research, when you have a quantitative study, the standard things you're going to see are table 1 is an explanation of who did the research, and that should be for qual or quant. So the first section of your results, whether it's, table 1 or there's a table earlier, either way, first part of the results must be a table of your participants, or depending on the sample, maybe they put it as a description, but that's quite rare. I would expect to see a table telling me who completed this study, how many of these people, how many of those people. The next thing you'll see is frequencies. So we measured the Maslach burnout inventory, we measured the Connor-Davidson resilience scale, and we measured the quality of life scale. So I want to see how many people scored each score on those scales. So you're not telling me about how they're related, but just what numbers did people provide that you analyzed. That's table 2. Table 3 is what are the correlations or the relationships between these variables. So again, when we have a positive correlation, it means both variables go in the same direction. If one goes up, the other goes up. If one goes down, the other goes down. That's considered positive. If we have a negative correlation, it means there's an inverse relationship. So one goes up, the other goes down. The temperature goes down, and some people, their going outside goes up because they know they can go skiing. That would be a negative correlation. So we want to look at the correlations. So we're probably going to find that quality of life has a positive correlation with resilience. Quality of life has a negative correlation with burnout, and resilience and burnout have a negative correlation. So it would logically make sense that if your resilience score is high, your burnout score is probably going to be low. Finally, table 4 is our regression analysis, and this looks at the nuance of how the different variables are connected. So this will tell us, okay, high resilience scores predict higher quality of life. So this doesn't just tell us the nature of the relationship, one goes up, the other goes down. It tells us like exactly how much does this influence the other variable. So you might be able to say resilience is related to higher quality of life, and it causes about 10% of the effect. Okay, so then we know that about 10% of if we're trying to boost someone's quality of life, 10% of that factor is related to their resilience. So maybe their resilience is something we want to work on. So we see this quite often with studies of resilience is they'll come back and say, okay, nurses coping in this environment, 8% of it is predicted by resilience. So we're going to give nurses resilience training. Well, if we did that study, we'll probably also find that the quality of management predicts about 20% of nurses' quality of life. So when we know the relative proportions in those predictive relationships, we can say, hey, yeah, there's part of this is due to personal resilience, but we're going to get way more bang for our buck if we focus on supporting the nurse managers and giving them leadership training, because that will get us way more influence because that constitutes 20% of the outcome. So it can help you decide like what are the priorities for intervention and how much of this phenomenon do we really understand. If you've only got 3 or 4% of an understanding of the effect, you're probably not going to want to do interventions because it's such a small piece, you're missing something. So you say, okay, we need to go back to the hypothesis level. What is really contributing to nurse quality of life so that we can get a bigger chunk, understand a bigger chunk of it, and then figure out how we're going to intervene there. So within each of these different pieces, we have different designs. And so the next video is going to go through what all the different research designs are, but across all of them, a lot of these first principles are quite consistent. So the samples are generally large, whether you're doing a randomized controlled trial or a prevalence study. You generally use surveys or some type of objective data collection that's verifiable, whether you are doing a quasi-experimental design or a correlational design. And so now we'll stop here and then I'll go into the different types of designs in more detail.